Hi, my name is Alex Fisher and I'm applying for the Cappel Scholarship. Now, I've been in love with life sciences for a while. Each morning in middle school, I would actually watch science videos on YouTube while I waited for the bus. Channels like SciShow and Life Noggin were my favorite, and they always had something new and interesting posted about topics like chemistry, biology, scientific discoveries, and so on. I like them so much because they always ask questions that I hadn't really thought of before. How exactly does Tylenol work? Do new memories replace old ones? Why do we give dogs tick medication but not humans? At the time, I watched these videos just as something I could do to pass the time in the morning, but now I really see it as the start in my interest in science. Once I was in high school and I had to start signing up for elective classes, I noticed a new class in my school's course catalog named Principles of Biomedical Science, or PBS. The course description talked all about how it covered subjects from forensics and anatomy to microbiology and biochemistry. I immediately enrolled, and I'm glad I did, because PBS became one of the most fascinating classes I've taken. So I can say with confidence that this class alone strongly convinced me to pursue a career in life sciences, particularly in biochemistry and microbiology. Now, what does any of this have to do with my favorite scientific discovery? Well, my time watching all those YouTube videos in middle school, I heard about this thing called CRISPR every once in a while. It was a very complex idea for me at the time, so I didn't fully understand it. But in high school, I kept hearing about these new discoveries and breakthrough in this CRISPR technology. So I became really curious about it and started looking into it, and what I learned was fascinating, enough to make it by far my favorite scientific discovery in life sciences, or any field of science for that matter. So, CRISPR is a set of DNA sequences in bacteria genomes that come from previous bacteriophage infections. Now, on the rare occasion that a bacteria cell survives a virus infection, these sequences help the bacteria identify and eliminate DNA from similar bacteriophages in future infections. This system effectively acts as an immune system for the bacteria, similar to how our body's immune systems can recognize a similar pathogen and quickly eliminate it before it does much damage. Now, scientists have actually known about this system since the 80s, but more recently, a group of scientists discovered that the CRISPR system can be used as something comparable to like a copy and paste tool for genetic information. It allows scientists to easily take genetic information, either synthesized or from another organism, and insert it directly into another organism, even of a completely different species. This is groundbreaking because with the ability to manipulate genetic information, theoretically we will soon have the ability to cure countless genetic diseases, from the inconvenient ones to those that are life-threatening. These include color blindness, trisomy, cystic fibrosis, sickle cell, hemophilia, Huntington's disease, and more. Many scientists also believe that CRISPR could even be used to strengthen a cancer patient's immune cells to become better cancer cell hunters. Now, of course, there are huge ethical concerns that need to be considered, all that are inherently to come with a technology like this, especially when we're talking about modifying and manipulating human DNA. But in my personal opinion, that shouldn't dissuade us from developing this or any technology. In fact, right now it's standard in many places around the world to test for dozens of genetic diseases in prenatal babies. And sadly, it isn't uncommon for the presence of a genetic disease in the unborn baby to result in a pregnancy termination. For example, Down syndrome, or trisomy 21, is the most common genetic disorder in babies, and in Europe, 92% of all pregnancies where Down syndrome is detected in the baby are terminated. It's heartbreaking statistics like these that inspire me to promote a reality where CRISPR may soon give us the ability to dramatically drop this number to zero, and I've made it my mission to follow a career path in life sciences so that I can help progress this technology that has such a powerful potential in the world of medicine. Thank you for listening and for your consideration.